Yes, we can hear you.
Hello, I'm gonna tell you about OBIS and genetic data today. So I'm Saara Suominen, and I work for OBIS as the scientific officer for genetic data. And one of my jobs is really to work on the eDNA projects that we have, but also try to figure out how we integrate eDNA data and talk with the community to understand what's needed, what are the main issues, and try to figure those out with all the stakeholders and everybody that's involved here. So today I'll give you a little introduction to what is OBIS and the data standards that we use. I was asking around, so some people seem to have a good idea and some don't. So I'll just give a short introduction to what Darwin Core is. And then we'll do a little bit about how the genetic data is added, and then we'll go into the hands-on training. And so I've shared the GitHub repository so also for everybody online, you can already start with that. And then we'll do some workshop calls. We can have our studio. Are there any questions? So, as I said, I'll talk to you about OBIS, about how we work with data, how data comes into OBIS, and is shown there. Then the data standards that we use for genetic data, and just shortly about data publication. So OBIS is a marine biodiversity database. We provide free, accessible, and detailed marine data. We have over 100 million current records moments, and of this, over 24 million sequences already. And we haven't been taking sequences for a very long time. We don't have that many data sets yet. So this is, I think, showing how much potential there really is in the data to bring a lot of information into our databases and to our biodiversity fields. OBIS is about 20 years old. We started with the Census of Marine Life, and we are part of IOC, and we are recognized as a global open access data and information clearinghouse on ocean biodiversity. So that means that data that is in OBIS supports many global policy assessments, for example, the World Ocean Assessments, IPBES assessments, and other science assessments for science-based decision making. So the motivation to put your data in OBIS, it goes directly into these processes. So it's really important that you share your data. We are a permanent program component of IOD. So that's the International Oceanographic Data and Information Exchange, which is part of the IOC. And there are several programs that are part of IOC. We heard about OBIS a couple of days ago from PhD. And we also have the Ocean Teacher Program, online training platform for all kinds of open access marine training. So how we work with data. We collect data from all kinds of different sources. So it can be articles, it can be long-term monitoring, it can be small studies, it can sometimes be citizen science. And this data goes through regional and thematic nodes. We have about 30 nodes around the world. So the nodes are the ones who are responsible for taking that data and formatting it and adding it to OBIS. So if you do have data, I'll give you a list of the nodes, and you can connect with your node and see what's the best way to go forward. And then that clean data and formatted data is integrated into OBIS. And through OBIS, it's fully open access. You can access it through different methods. So we have applications, we have an R package, and we have a visual mapper for people to access that data. And it's also very easy to build something on top of OBIS. So if you do want a custom-made application for using OBIS data, then that's completely possible. We can figure that out. So here's a list of our nodes. You can see we have around 30 nodes. You can see we have quite a bit. So the idea is that we provide long-term sustainable storage of biodiversity data. It's machine readable with all relevant data possible to add to OBIS. And that includes measurements of environmental parameters, so it's not only biodiversity information. And we want to provide reliable data that can be compared. So of course, we adhere to the FAIR principles, and that's why we have the data formats that we use that are shared around the world. And that's why it can sometimes be a little bit difficult and complicated, but it's really worth the effort also to get into that data standard and share your data, because this is really a community standard. What it means that it's a standard format means that there's universal and comparable language use, which makes it interoperable and machine-readable. And of course, there's all the contextual information. We use Darwin Core archives, which has different tables. I'll talk about that. So what is the Darwin Core archive? Or the Darwin Core, it's a body of standards that facilitate sharing of biodiversity information. And that includes identifiers, labels, and definitions. So everything should be well defined, what is used in the Darwin Core standard. In a very simple form, it works so that you have a core table. Basically, that can be a CSV table, an Excel table, where you have most of your information on your species occurrence. We work with species occurrence, location and time information. And then depending on how your data was collected or what the structure is of your data, there's different extension tables that you can add that will give that ability for you to record that structure or record those special things about your data. More DNA data. There are very specific fields that you can add for the information. Importantly, there's stable terms and vocabularies. And this can also be quite complicated, but it's really, this is what makes the data interoperable and machine readable, which means you can go back. And then if you have data from other parts of the world, you can go back to that information. This is maintained by the TADWIG, Biodiversity Information Standards for Native. So it's really not only used by OBIS, but it's used by GBIF and around the biodiversity. So we have currently 8 required Darwin Core terms. I'm going to go into those in the next slides. Something special about OBIS is we have a taxonomic backbone, which is the World Register of Marine Species. So all your taxonomic names should have an ID from the backbone. And that again is something that keeps things comparable at another level. Now this can cause quite a bit of issues with genetic data because we are not so well defined always. We are not so well linked to voucher specimens necessarily all the time. And we also have all kinds of unknowns, right, that pop up. But there are some kind of workarounds that you can do to still record all your information, especially all your DNA sequences in your data sets while having those names that you can and then other names that you cannot yet have. We also do quality checks on coordinates and depth information, and that specifically will look into if your data is marine data and that the depth is reasonable. And we can record environmental information with extended measurement or fact extension. So these are the required OBIS Darwin Core terms. It's not a lot, so you can add information, very minimal information. Of course, this is not what OBIS is for. We want to be able to have all the contextual information that is needed for data reusability. But basically, we look at events, so the dates of where you took your sample, the locations, your occurrence, the basis of record, and of course the name of the species that you adopted.
What does it mean, basis of record?
I will get to that in a bit. Yeah, it's just a, it's a category. So there's very specific categories that you can add to that or choose that there. But yeah, so dates needs to be a specific format, and we do check for that. That's quite simple to fix. And the latitude and longitude need to be in degrees, in decimal degrees. The occurrence status must be present or absent, so we do have some absence records. And then so basis of record will be one of the following. And so of course we differentiate between specimens and human observations. Or usually we use material samples, so that's especially for DNA samples because we do have a physical sample where the data is coming from. And here you can link your IDs from the collection, whatever is important for you and your data sets to understand which. And then we have scientific name, again, we strongly recommend to include the scientific name ID from the World Taxonomic Database. OBIS does also search for scientific name IDs if you don't provide them, but of course it's better if you already work on that yourself because there is some. So OBIS does it when it ingests the data, it does it quite automatically, so there are issues there that you may need to solve. So how it all comes together is by putting these data files into a Darwin Core archive, as we say. So that's very simply just having those core and extension tables and a few other metadata files and putting that into a zip archive. And that's what we mean when we say Darwin Core archive. It's a very simple concept, but it's something we use all the time. So for the core files, we have two different types. So there's either occurrence core or event core. An occurrence core is when you have just a list of species and you have the information related to it. Event core you can use, for example, if you want to have your crews linked to sub-events, linked to subsamples, linked to species. The thing is now, at this moment, with eDNA data, we can only use occurrence core because it's an eDNA data extension. So we can only have one core extension relationship at the moment, but there is a change to this. And it will be incorporated by the end of this year, according to our data manager, Peter Provost. So, which means that you will be able to have an event core, an occurrence extension, and a DNA-derived data extension. So it gives you a bit more flexibility in the data structuring. But yes, so extension tables connect additional information to the core table. So that could be the occurrence extension if you're using an event core. Extended measurements or facts. So that's where you want to add, for example, chemical measurements. You can see that here. So it gives you the ability to add any kind of measurements, any kind of value, any kind of units. But as you'll see in a bit, you also add IDs to that to keep that vocabulary. And then we have the DNA-derived data extension, which is what we're going to use and talk about today. So in the future, in your DNA-derived data extension, you can have an ID that links to your event core, and then you can also have the occurrence ID, which links to your occurrence extension. So it kind of makes a multi relationship. And so just a quick note on the extended measurement or fact table. So you can record any type of measurements. So again, you have your measurement type, measurement value, and measurement units. But the thing is, the measurement type is free text. It means that you can have And we know you have all kinds of different ways of saying, okay, the length of your fish species. There's all kinds of different lengths, there's different ways of saying the length. And therefore, we also use identifiers for each of these columns, which can then standardize that field that you want to add to your data. And for this, we have vocabularies. And this is a whole different field of complexity, but it's, I think once you have your studies, you know what types of things you're measuring, then you can figure out what the vocabulary IDs are for that. And then it's, of course, a standard workflow, which will then help us to have that comparable. I'm not going to go into it, but if you are interested and you want more information on how these work, how to choose vocabularies, we have Elizabeth at the Secretariat who's looking into this a lot. There are several videos online as well to figure out how to do that, or you can contact us. So the DNA-derived data extension, as I said, now we use the occurrence core and the extension. We have a guideline for that, which is published together with GBIF. So all the information that you need is there. We also have a very comprehensive OBIS manual that you can access through our website. And there you have in detail all of these different steps that need to be done for eDNA data, but for all kinds of information. Steps that you need to do for publishing this. Of course, DNA data is special in terms that it's derived, it's not direct observation. It means that we have a lot of steps, we have a lot of things that we would like to understand when we look at the data again to say, okay, how did they do this? What happened in between? There's also a lot of unknowns. We are aware of that, and I think the strength that we have in having a database like this is that we can record that sequence. We can record those unknown sequences, which means we can go back to it and say, okay, now we understand better what this is, what category this is, or even maybe what species this is in the future. So just to go over a bit the key metadata that you would have in an eDNA metabarcoding study. So here in green, you have the step of the workflow. In yellow, you have the different types of metadata that you would have linked to that step. And in gray is the terms that you would have in the Darwin core. In the occurrence core table are the ones that are in black. And in red, you have the terms that are in the extension table. So for example, for the field sampling, you can add the environment broad scale, which is also something that is in the MixS. The MixS standards. So most of these are the DNA derived data extension was built from kind of taking things from the MixS standard, as well as other things that were necessary. And as I've been talking this week, there's also a lot of developments that can still be done, and we are still doing and want to do with also with Miwa's work on fair eDNA. So these things will develop. But you can add information on the DNA extraction methods, as well as on the markers that you use for amplifying PCR that you've done. And again, the sequencing, still the raw sequences need to be submitted to a public repository, but you can link that ID to your data sets with the associated sequences. And then finally, after your analysis, you will bring together those different tables into your final information with your annotated species, as well as your sequence. Yes.
Is the red necessary?
There are recommended fields.
Okay.
But there's no, so you can have just an occurrence core, but the whole idea is that with the DNA extension, you add your DNA sequence. And then whatever information you want to include in that to make sure that it's like understandable later on if you come back.
If you don't have your absorbance to 60-30 ratio.
Yeah, you don't need to, but if you have it and you want to
nice.
share it, you can. Yeah, no, there's the 8 required terms, and that's pretty much it. after that, it's up to you as to how much you want to add. Of course, you should add everything. You can also add a link, of course, to a publication or a standard operating procedure or something like that. But again, the more you kind of write out everything in different categories or different columns, the more you can then search on that data and kind of like compare data across different fields.
This is for an occurrence core. And so for every sample that you submit, every set of observations, you need to submit both an event and an occurrence core, or you kind of pick one, and for DNA it would have to be this one.
Yeah, exactly. So if you do wanna submit an event core, then you would use the occurrence extension to add your occurrence information. So your
Okay.
species information. would
Is that
be-
because an event can have multiple occurrences?
Yes, yeah,
It's like an event might be like a sampling expedition,
exactly.
and then each sample would be an occurrence.
Yeah, yeah. Yeah,
Is it, sorry, I guess I'm getting, is it each ASV is an occurrence?
yes.
Yeah, I think it's each ASV is an occurrence, and then with do you say the sample name under material sample ID?
Yes,
Okay. Okay, so instead of submitting like an ASV table for every sample, you might submit a sample table for every ASV, I guess, like you just kind of inverse where the ASVs occur in which samples.
if you use occurrence core, yeah, then you do long formats. So basically everything you have is in one row. All the information that you have is in one columns. So it does inflate the data quite a bit when you're using occurrence core because you're repeating everything. Which is in every sample.
Got it. I'm sure this will be much more clear when we actually do
Enjoy.
it, yeah.
Yeah, so we have different guidelines, manual. We have a webinar now that's a few years old already. We had a genetic data working group, we have a use case from Monterey Bay. And of course, now we have this training. So we have a lot of materials for when you want to add your data or format your data. And of course, you can always contact us with questions and to discuss issues that you have because it does, when you first get into it, it does take some time to figure it out. And this is the main guide that you should refer to. And we say that OBIS is and it can be an ASV repository. So we have all the setup ready for having fair data. We're already a very established database and eDNA actually fits quite well into that. We have a lot of guides for that. We are developing different data products from that eDNA data. So we have the decision support tool, the dashboard that I've also mentioned related to our projects now. And we're also involved in many European projects for eDNA data standardization. So we're kind of involved in all the different steps of the eDNA workflow. And we understand how that metadata from those different steps has to come into the data. And how that influences what we do with the data. So for example, we're working a lot with looking at the probability of species annotations that I think Eric was also talking about. And the confidence of different annotations. So all of these things are in the process. And we are open to collaboration and working together. So just a quick note of how OBIS data publication usually works. This is actually what I also talked about last year. I'm just gonna have the first few slides that I had last year on this. We use the Pacman pipeline, so it's a bioinformatics pipeline that's developed for the Pacman project. And basically this pipeline formats the data already to a Darwin form. That means that it simplifies that step, right? So that's kind of an easy way to do that is that you get out your tables directly from your bioinformatic pipeline. It stops you from having to do a manual formatting of your data. So we get an occurrence table and a DNA extension table directly out of that. But if we look at how we traditionally work with sequence data and metabarcoding data is usually you have these different files and different tables. Okay, if you work with phyloseq, you know that there's these slots for everything in that data format. So you have your taxonomy table, your sample information table, your OTU table, and then your FASTA file for your sequences. So what you need to do to publish that in OBIS and in Darwin Core, as I said, put everything together. In phyloseq, you can do that, right? There is a pivot longer or whatever it's called, PS melt, I think, to get everything in the long format. So that's simply what we do is we link all that information. So for OBIS data publication, we have an IPT, which is the Integrated Publishing Toolkit, which is the same that GBIF uses. So that means that you upload your data sets there. Usually you use your nodes IPT, you upload your data sets. And through this system, you map your field names to make sure that they're correct and add your metadata. So just here showing the different tables that we had the current score, and this is the DNA extension. Uploaded. Again, we have everything in the manual for you. But basically when you're in IPT, you just go for create a new resource and you go for that. So I just wanted to show that that's the normal way of doing things. You upload your tables and it maps those fields for you, and then you publish that. But now we're going to do a bit more manually this thing. So we're going to go into the hands-on training and run through a test data set. It's a small data set. It is 2 real samples from the eDNA Expeditions project. So it has all the issues that we might think. We're not going too deep into this, but we can look into any questions that you have. So maybe I can also put this in the chat, or does everybody online? Is that easy for you to go there?
I can do it. So. Photo if you need to move on. Okay.
All right. Any questions? Yes.
But can you like briefly say what the Pac-Man pipeline is or where that fits in? Like is that raw data to processed, or is that something people using to analyze data?
Yeah, it's a bioinformatics pipeline from raw data to process. We developed it for the Pac-Man project because there was not a standardized bioinformatics pipeline to be used, and I wanted to combine different things together. So that's the reason. And it's one of the ones we have in the comparison as well. It's not published yet, but it is on GitHub open access. So you can see also all the steps that are required for the formatting are of course there.
Does it incorporate data two and things like that? Or
Yeah,
is it
it's data
something-
two. Yeah, it's data two. Actually, this is not correct anymore, sorry. It is data two, and then we have a taxonomic assignment based on the RDP. So this is not correct. Don't take a picture of that. And then, so we actually do taxonomic assignment with RDP and with vSearch as well. So we get those vSearch identities for our references, and we get that reference sequence from the NCBI or whatever your reference IDs are. And we will do a blast as well. So we have kind of multiple different annotations, and then we try to figure out what it actually is.
And do you specify your reference database then, or is there?
Yes, yeah.
You incorporate any cleanup steps?
Clean up from the annotations.
Yeah, later on, yeah, so yeah,
Yes.
it's a bit
Yeah.
weird.
Yeah, it's not in the pipeline, so there's a lot of, well, we have one workflow now where Peter has looked at the distribution of the species versus where they are, so how far they are from their expected distribution and the thermal limits. So he gives kind of a quality score, but after that, we're going through them manually to see, okay, yeah. And we also, for Indian expeditions especially, we sent those lists to the local management for them to give us feedback as to which they expected was real and which definitely was not a real annotation. So this doesn't have a solution for us, but it's, yeah. I think it's important, yes.
How similar is it from GBIF, what GBIF is implementing? Is it this similar than Pacman, is the same or?
You mean for the annotations or?
I think you want to do a bioinformatics pipeline now, or they have it already?
I haven't heard that they're doing, but maybe they are, yeah. So that we'll go into the GBIF had this tool, the Metabar coding toolkit, where you could upload these types of tables that we have, so there's different tables, and then it does the mapping and formatting online, and it can do also a taxonomic annotation. But the tool is under development still, so it's not, but again, that's like, you have your ASVs, and then you just quickly look at what it could be in the reference database, so that's one way to do it, but still you need to clean up your data to get to your ASVs or your OTUs first, so it's not a full bioinformatic pipeline. I don't know if they have I haven't heard of that, but I wouldn't be surprised.
But that'll be very important to compare all of them,
Yeah, absolutely.
to at least know what's the difference, and if
Yeah.
we see a difference, how to make it.
Yeah, for the annotations.
Yeah, we probably have benefits and, like.
Yeah,
both,
indeed,
but
yeah.
To know that.
Yeah, so what we're trying to do in the data analysis challenge is so that's part of a European project where we look at the outputs of different bioinformatics pipelines and see how accurate they are. We have aquarium data, so see how accurately we get to species annotation. So if you have a bioinformatics pipeline that you want to compare,
Yes, we do.
you can join.
We do, and I think at least for us, OBIS is the official government Canada platform to prove this, so we have to go. So that'll be a very important between Canada, we have. Even within us, within our group, we have different pipelines, so maybe we can group that and provide that to you.
That exercise happening?
So it was, but since you are a special group of people, you can still join. So we have a few test data sets. So we're asking everybody just to analyze the same data set and give us these final tables. And then we're comparing the results. It's a very simple exercise. And what we were hoping is that because everybody uses their pipelines a little bit differently, maybe with different parameters, they have their favorite workflows or favorite little tweaks. So we're hoping to kind of get all those recorded instead of using pipelines with like just standard parameters, because then that's not how you're meant to do it, right? You're meant to really look into what you're doing. Yeah.
Are the results from that gonna be public? Like,
Yes.
and so if you say you felt like trying out a new bioinformatic pipeline, you could download test data and then see how it compares to what's, yeah,
That's the
interesting.
idea, yeah. Because at least for me, it was frustrating. I tested a few different pipelines, but you don't really know. And there's tests available, of course, people have published some tests, but it's like you do one specific data set or 3 different pipelines, and then you can see from those 3 what is better for your specific test workflow. Then, yeah, it's not a general. So my wish is that it becomes like this kind of living website where you can add and keep testing the same things.
Is the reference database also an option, or are we meant to use the same reference database
We're
too?
meant to use the same, so we also provide a reference database, but of course. Maybe in this, but people did it because we had one provided, but it wasn't so comprehensive. So we didn't have, for example, bacteria in the reference database. That was provided from our collaborators. So people actually did 2 annotations. Usually they did one with with the provided reference database, one with their own, so we're also gonna. Right, so. Go ahead. I'm gonna share my screen here. So now we have the GitHub repository. So this one has all of the different files you will need for your training. You can always go back to that. We have a very small eDNA data set. So that's 2 samples from the eDNA Expeditions project. Seychelles, without that, I call it. And in different form, like in the different tables that I explained earlier. So I don't know if you agree, but at least this is how we are usually working with eDNA data in these different tables. There is still a bit of information here on what the extension is, for example. And again, on the publishing guidelines that we have. So this is the main parts of the publishing guidelines. So if you haven't had a chance to look at that, basically it categorizes eDNA data into different types, which are explained here, and metabarcoding would be category. So you have sequences or like barcoding sequences, metabarcoding sequences linked to species information. It goes through the processing workflow, which is normal for a metabarcoding study or eDNA study, and then looks at data mapping for that. So there's some things that are a little bit different between OBIS and GBIF, and I already talked a bit about that. So that's especially the worms taxonomy is a very important difference between. So GBIF also recognizes the worms taxonomy, but it has also different taxonomies. So with GBIF, you can add, for example, NCBI taxonomy, but we haven't gone to that because we want the taxonomies to be really clear. So we have different ways you can add that into the different fields like, so verbatim identification, you can have whatever name you get from your annotation. So usually you have these types of species unknown, environmental, and little ID and something. And in taxon concept ID, you can always add those IDs as well, and NCBI ID. So everything is saved in your data set. It's just not in the scientific name. Well, actually, you can also have it in scientific name, but then your scientific name ID should be from worms. So for example, could be just bacteria or whatever else. And then our unknowns are recorded as inserte sedis, which is scientific name ID 12. You'll have a lot of that. And we have different recommendations for qPCR data. But we're not going to go into that now. If anybody does have qPCR data that they do want to add, then I would be happy to work together to. My next step is data sets. So as I've asked everybody to clone the repository. And then we have a existing solution, which is called, it's not in the solutions folder, it is here, peter.rmd. So that's provided by our data manager, Peter Provost. So what we're gonna do is we're gonna run through that. But if anybody does want to try on their own, that's completely possible. So then here are the different steps of what you would need to do for your data sets. So you want to determine what you want to do, want to combine the different tables, and then use the OBIS tools package to do different steps. So you can do the taxon matching, coordinate checking, checking the dates, checking other possible missing fields. You can create an extended measurement or fact table, and then create the DNA derived table, and then generate the Darwin Core archive. You compare, depending on how much time we have, but I think we probably should have plenty of time. We can use the GBIF tool. So this is what I was saying, the meta barcoding toolkit, and I prepared a small data set that we can upload and you can see how it works. So that would be here, dataset subset GBIF. And there's some small differences to what we provided that you need to do, but it's quite straightforward. There just needs to be a column named ID and so on. And I've added the taxonomic levels for that because GBIF shows that then. And then we can also check our data set using email. All right, are we ready? Yeah.
Yes.
Don't know if anybody, I guess nobody ran through everything yet, right? So.
Does everyone have what they need if they're not online? We've got some spare laptops that do have internet connection if they're not.
I think it's hard to know until we
Yeah, start. See,
Yeah, so we can just maybe first look at the data sets. So we have these tables for the data sets. So the main ones, of course, are the D table. We have 2 samples across, I think, around 15,000 ASVs. And there's a lot of ASVs. We have the sequences that are linked to these ASVs in a FASTA file. We have taxonomy, but this is only the scientific name. So this is the final taxonomy. We don't have taxonomic levels for that. And then we have very simple sample data. So that would be event dates, the area, the location, the depth, and DNA extraction amounts. So we're gonna put this together in simple combination. So I think, is everybody here in our studio? All the libraries are loaded. So I'm happy to run this together, or if people want, you can start running and we can do questions. Meanwhile,
So we should open the Peter.
yeah, peter.rmd, easiest in an RStudio instance, or whatever. you want to use.
And change the output directory to.
Change that, yes. To your own name, and you can, I mean, you can copy Peter and rename it so that you change it somewhere else, but that's okay.
It says the package worms is required.
Yes,
Apparently I can install packages, so
you can install,
yeah.
okay, good.
Oh, geez.
everybody in our studio. And you can set your working directory. So you might need something in the
So it looks like there are two samples.
two samples, yeah.
Are there plans to maybe include an example or quick script to get you from your normal taxonomy table to the single column taxonomy, or just wondering, yeah, if people had NAs or some common term for unknown across their taxonomy table, if you guys would provide like a script to help it make it easier for users to just
To go from.
the usual, the output of common bioinformatic pipelines like Chime or other things to the single column,
Yeah,
I guess.
yeah, single column, you mean the scientific name, just that we have here.
Yeah,
Yeah, yeah, absolutely. We can. So what we have here is just the highest level that we could annotate. So there's a lot of eukaryote, right? So a lot of these are unknowns. So yeah, it's just, we just take the first one that's not an A.
And that has to be Worm's taxonomy.
So at this stage, it's just a name that we get pipeline, and we're gonna match that to the worms. We're gonna match
But-
those names here.
Right, but it has to exist in the Worms taxonomy.
Yeah.
Like you can't have an identifier that is not in Worms, because you can't make that match otherwise.
Indeed,
So
but yeah,
when you do your taxonomic annotation in your bioinformatic pipeline, you have to make sure that you're annotating with.
well, I mean, worms is scientific names, so whatever you annotate to a scientific name, usually you can find in worms. There are
Yeah,
cases.
but like with NCBI, they're different.
Indeed, yeah, but of course, like. So, for example, for bacteria, it's very messy, right? You have all kinds of different IDs and indications, so. What we now say is that you can keep that name, scientific name. And then the scientific name ID will be whatever higher level you do know, so that would be bacteria for example. So there are ways to keep all that information, or in verbatim identification. That you can, and taxon concept ID can be your NCBI ID, so there's many ways to, you're not gonna lose any information. But it's just that our scientific name ID is an AFIA ID.
This may be one of Catherine Sullivan's tools, I think maybe is to link NCBI Worm taxonomies.
I think there's Worms lookup tools. So like, which I think might be one of these things where it's like you give a list of names and it gives you the worms if there's a worms match. I think it's just difficult if there isn't a worms match, then you have to bump it up, like you were saying, to like the highest thing that has a worms match.
Got it.
And I don't know enough about worms, and, like, we do like terrestrial things in our data set, which, and so it's like deciding, do you filter those out and not submit those to OBIS? And yeah.
So OBIS will not show those that are not, but they're not a problem, so they will be in the original data sets, so. So your data will be there, but it's just not shown. And it will be flagged. So, has everybody been able to list their files? Right. So then we can start reading the different tables. And we can see them again, so you could come out. Yes.
With the organism quantity,
Yes,
these are just your that will be highly dependent on how deep you sequence, right?
yes,
So
yeah,
that number is pretty arbitrary.
yeah,
Doesn't
yeah,
really mean anything.
no, yeah, so we have. Is it organism counts? So there's sample size. Sample size is the total amount of reads you have in your sample.
Okay, so yeah, you have good of them.
Yeah, and then you have a category for that, which is DNA sequence reads. And then you have the organism quantity, which is then the amount of reads for your ASV. So you can get to your relative levels. Taxonomy file, same thing. They're just simple tables. You read your Excel file.
It seems like too, like for species, if you have a two name species, you have to put an underscore in there. Or more, maybe.
No, we're actually gonna remove those under.
Oh, okay.
It's one step of our process, yeah. Okay, so then we have our sample table, our taxonomy table, and our OTU table. And we're gonna combine those. Sorry, joining the tables, well, first we have the event fields. So this will be formatting basically the sample information into Darwin Core fields. So we're simply doing selection and renaming of those fields. So you can see we have event ID and material sample ID, that's the name of our sample. Both of them. But again, this depends on your data set and what you're doing, what you want to have there. And in this case, that's what we do, yeah.
And just to clarify, should your material sample ID be like, for example, the name of the DNA extract or the name of like the sequence sample? So like, should it have the same name if you do multiple barcodes or sequence something multiple times? Is it the same material sample ID?
In that case, I think it would be
Like
a sample.
Your-
So whatever, right, whatever you have.
Every time you sequence something,
Based on
material sample.
Yeah, depending on how you want, right, what makes sense for your. For your institution, for how you store your name. What is the best way for you? What is the?
I think it is useful to be able to link back to this came from the same water sample or
Yeah.
this came from the same filter from the same water sample.
Yeah,
So I think having that in there somewhere is very useful, but it's just thinking about maybe where you wanna put it versus,
yeah,
understanding that
yeah,
separate sequencing reaction or this.
Yeah. trying to think, so for events, cause then you could link to the same event, right? You could have your material sample ID and event ID, or like your sample in your event ID, and then your material sample ID can be your actual extract material.
But you might, well. But what if you're sampling at one site, but you take multiple samples? So is your event the sampling event, or is it your
So you can have parent events.
parent's event?
Yeah, great. So you can do all these things.
Okay.
yeah. Right now, it's not so easy because we're in the occurrence core. But once we get into the event core, you can do all these things. So here it's really, as you see, it's kind of mapping the names to the Darwin Core names. So again, these were the names, so name, event begin, area name, and so forth, were the names that we had in our sample table. And we're just finding the terms in the Darwin Core which fit to those names that we had in our table and rename. And that's basically what it means to map to Darwin Core.
But it doesn't matter what headers you have in your metadata file, or is this hard coded?
No, it doesn't matter.
Yeah,
No,
right.
you just, you could do as you like, you have your table with your names and you map them to the Darwin Core.
Okay, but I don't have to alter the script. Sorry, that's what I mean. So
If you, here,
Yeah,
no, like
no.
for this example, no, it's fine. I haven't done any little
okay.
tricks here for
Yeah.
you.
But if
But if
your
you-
metadata table had
It's
like a
different.
column called
Yeah.
latitude,
Then I have
then
to
you
add-
have to change that
Yeah,
to
yeah,
latitude,
yeah.
for
So
example.
if you say, I don't know, location longitude or something, and then you name it again to decimal longitude. It's just putting those terms, right? Are there any questions on these terms or how to find other terms or these kinds of things? Okay. Again, there's a lot of resources online for using Darwin Core. You can always go to the OBIS manual to find it.
We have a question, is there someone that we can reach out to this directly?
Yes, of course, yeah. So you can, You can reach out to your nodes if you want, Canadian nodes. Or you can reach out to the secretariat as well. So now we're looking at the occurrence core table. Now, we have in eDNA data, we have that format of we have ASVs as rows and samples as columns. So what we need to do is just make that into a long format. So this is what's happening here is that we take the sec tab or the OTU tab and we just format it to long format. So maybe important to note is because it's a long format, we need an occurrence ID that separates each ASV found in each sample. Right, so you will have an ASV in your ASV table, you will have an ASV across multiple samples. So each of these becomes a separate row. So you have to have an occurrence sample and an ASV identification.
It's also handy that you can change the filter greater than how many reads.
Yeah, in this case, we're just removing all that are zero, right? So if you have, you will, in your ASV table, you will have like, in one sample, you have 14 reads, in another you have 0. So because that also becomes an occurrence, we just remove the 0 occurrences because they're meaningless in this case, because we're not recording absence rates.
And is here where you'd also create your DNA sequence reads column of number of reads per sample, or will we be doing that later? Or
That will be in the, that will be in here, yes.
organism quantities number of reads per ASV in
Right,
each sample,
right.
and then you also want total reads. per
Oh,
sample.
I see.
You can have the taxonomy. Is it not working?
For me.
Taxonomy must be numeric, not a data frame. Works for you? Yeah, okay, it's fine. Right.
So here we renamed the taxonomy column, which was our scientific name, our highest taxonomic level, to verbatim identification. So this is a field that will stay, whatever you do, this will stay the same. We'll also do kind of a legacy field. And we need, of course, the ASV as a ID. And then we join to each ASV, we join that taxonomy information. Comes here. Data identification. And then we will join the information we got from the sample table. That's what we mean by events and occurrence fields. Join all of those, and now we have our sample information, all that event information related to that, so location, depth, sample size value, and so on. And our information on the ASVs and taxonomy in one table. It's very simple formatting.
Right now, DNA is DNA concentration. Is that
Yeah, so DNA is, yeah, indeed. It's the concentration of the extracted data. And we'll show you how it goes into the EMOF table, extended measurement facts. And then we can add our protocols, the link to our protocols in Bebop. So I think now we should be using something else, right? For the protocol.
Either GitHub or protocols.io. we got
Okay.
them.
Both are fine.
Yeah.
So you can add links to your SOPs, to your maybe published article. So all these that link the information of what you've done can be added to the table. So that's always. Yeah. So then we go into the taxon matching. This is done with the OBIS rules package. So first we remove those underscores. We get to just the list of names. And then we can use OBIS tools to match those names. Now this requires internet, so if you cannot, there is a table in the solution which already has those matches.
And I think also I got the error that I don't have the package called OBIS tools. So I think just adding it to the GitHub site. So I ran the little GitHub thing and it's not included, I think, in the.
Yeah.
Sorry, it's okay.
What's the best way to install
Or
OBIS
not.
tools? I'm gonna have to Google.
Sometimes just go to tool, install packages, and they search.
Oh.
Install packages.
Yeah.
Might be the same as install.
Not available for this version of R.
Yeah, so
It's the GitHub.
Where's the matched file that we can import?
It should be in the solutions folder.
It's commented. Matched result. Nice. Oh, I see. Yeah, you already have a line in there. Great. So the package, if we could run it, it's basically querying our name list and then getting the kind of worms taxon ID.
Yeah, exactly. So you can see it's linking the scientific name ID to your scientific name.
Does it also spit out ones that don't have matches? Okay.
So first, we add that to our occurrence table, so now we have a scientific name ID here. Yeah, and then we can look at non-matches. So those occurrences that did not have a scientific name ID, we write that out in a separate table as well. After that. But basically we can look, so we have a lot of eukaryote. So worms does not have a eukaryote identification. So these are gonna be unknown for us. We'll do that in the next step. But then you can see also the rest that did not have any matches. So many of these are undeaf, Nichia bones. And then there are some other ones that's.
Yeah, because we need to harvest.
Oh, yes. Maybe we want to have OBIS flag these and can also send an indication to worms that they're not found. But it's faster to do that yourself, so if there's something that you that should be in worms, you can catch those. But this is the step where it is a little bit manual.
So in theory, this could be, yeah, the bottleneck of your.
Yeah, exactly. Is everybody following for the library?
Yeah, I got a little bit tripped up with
Yeah.
the, so sorry, just to repeat, the OBIS tools package. Where can we find that?
Yeah, sorry, I should have, that's my mistake. So it's on GitHub,
Okay,
but I don't know if you have access here to GitHub.
Oh, there's.
One that it's just hashed out.
The solutions files are,
In the, yeah, you can just get rid of the hashtag to load the matched file, right?
exactly, yeah, you can get the solutions file so you can continue, but.
Okay.
Then in the next step, we are just formatting those eukaryota things to unknowns. If we look at now the non-matches. So we had here almost 10,000 of eukaryota. Now if we look at it, they're gone. Because we added the unknown scientific name ID, so now we have a much more manageable number of unknown names. There's more undeaf eukaryota that could be included.
My non-matches table empty.
And I also got an empty table, I think, just because we uploaded the solutions. When we worked from the solutions file, I think it had everything fixed already.
Yeah, right, yeah, I think I added probably.
Oh, the match table had already been fixed.
Has probably the unknowns already.
Got it.
But, yeah.
Okay.
And then I've added here a way for you to add different taxonomic levels, so that goes to the worms ID and looks up all the higher taxonomic levels. This is not necessary. You don't need to do this to make your Darwin-4 archive, or because these are linked in the database directly. But I did this because I had to add them for the GBIF tool, and I thought it's nice for everybody to see how that's done in case you want to look up higher taxonomic levels, for example.
That's a really useful tool.
Yeah. But it also takes some time because it does the worms, WM record is what we're using. That's the to get to those higher taxonomy.
Is that a file we have locally or does it require? Don't think we have it.
This one we probably don't.
Oh,
have, sorry. But we did, normally we don't need it, so I'll show you that. Basically, what it does is you add all of these levels, phylum, class, order, and a rank.
It's working.
Yeah.
Yeah, it just takes a while.
So you had the worms package.
I downloaded, yeah, this morning.
Everything clear so far?
Yeah, but is there a way to use like the phyloseq kind of object which pretty much has all of this already?
Yeah, yeah. So I've done that in a few cases, just a matter of reformatting, right? You get
Yeah.
the table separate, or you do a long format and you map the fields. Everything is possible, right? So you can do, it's not difficult. Maybe we can ask PhiloSig to add a function, export star and core, for example. That
Just,
could
I'm
be.
thinking more so like people will already have a file seek object for their analyses.
Yeah,
So you're cutting this whole step, so you don't have to do this,
yeah,
what you're doing. Then you already have your object for your ecological interpretations, and then you go like export to Darwin core.
yeah,
So you have your metadata, your taxonomy, your like phylogenetic, what we're doing now,
yeah, exactly.
it's already all in there.
Yeah, indeed, yeah. Yeah, it's the same thing, right? Because now we start with these separate tables, but it's the same thing. You can just refer to those tables in your PhiloSig object.
You still have to like rename your fields and make
Sure.
sure they map, and that's what this is doing. So whatever we were talking about, like exporting to this, be doing all of this behind the scenes.
I think it is a good point though, that like developing tools that would go from a file seek for the user who doesn't know how to do this maybe as much, like if they went from common things like a file seek object or a chime object and just said, here's a tool that'll take these common formats and
Yeah.
give you that, might be useful.
Yeah, is everybody using PhiloSig?
Really?
But I can get myself into
Yeah,
file seek,
yeah.
yeah.
It's not
Yeah, no, but
recently,
Yeah.
it's just one, I'm just
Right.
trying to think.
do a lot of base art stuff.
Yeah.
with
So-
my-
So we do a lot of this, so I do a lot of this.
Yeah.
Anyway, but
Oh, wow.
I think having, for new users, I think having like, these are common PhiloSeq or Chime or formats that people might have
Yeah.
already would be kind of useful. Or if the GBIF importer tool will take those tables anyway,
Yeah.
just work with the GBIF importer tool
Yeah.
and make sure it aligns with what OBIS
Exactly,
wants.
yeah. I mean, I think we could just make a PhiloSig, export PhiloSig in OBIS tools, for example.
And within the Chime ecosystem, there's also a way of you just creating a new tool within Chime as well.
Yeah.
So that's also another
That's
option.
actually.
Why we don't-
Just link to those existing.
Maybe there's some things next year we shall do during the workshop where we just like show in our lab what we found. The full step actually, how like our protocols at the point and like what we our new change and what we found the best and really like the very basic until great application. So like if for you use GBIF before changing this or you have that templates and
I was thinking as we work through this, what might be the most useful too is going through a workflow of, you always have to put your raw data in SRA. So like mapping out, how did the NCBI like biosample map to what OBIS considers a sample, or, you know, just having like an exact easy mapping. So you're like, okay, I have to get my data into this format. How do I move now to this one?
Yeah.
so like before the workshop, maybe we can have like all of us putting our workflow like from scratch and see what's the difference. So at least it's a very basic way to move towards optimization.
Do you know if Tara Oceans puts everything on OBIS? Probably,
Think so.
because they got a really nice SRA kind of like hierarchy for all the
Mm.
different samples.
Yeah.
Might be nice.
That's supposed to be.
Yeah.
Are you
But
connected
they tend not to
to
come to the meetings.
OBIS now is connected to Ecotaxa as well for imaging or no? Because I know that they were supposed to be published directly from Ecotaxa. That's imaging, but that's the same. Know if it's ready?
I'm not sure, but I will ask Peter.
For me, all of my metadata or for both, always do imaging and water EDA. It's an easy way to
I can actually.
double.
I think I can invite Peter here. Okay, anyway, are we okay to go forward? Am I going too slow? You wanna go a bit faster? No, okay. So that was basically putting the tables together, and now we're gonna do a little bit the quality control steps. Just so you see what happens and how OBIS controls those things. So first we look at the coordinates that we have. And lo and behold, they are on land. So something has gone wrong. So you can see here.
Oh,
Like
meanwhile,
palm
we don't
oil
have
now.
any-
Unfortunately, my leaf is.
Well, my code's just not working. Locality not found. Where's the-
At the bottom, next of it.
Yeah, page table.
Page table.
It could be because you didn't manage to combine with them.
I got the paged occurrence table.
So the occurrences we've been kind of adding.
Right, so this one has all the scientific ranks now, including all the metadata. Okay, let's see, scientific names.
You may need to do the event steps again.
Oh, okay. Event variable.
If you run something twice.
Oh, I see. That's possible.
So a common issue that we often have is one of the coordinates is the wrong way around, right? So this is what's happened here as well. So we need to flip that is what we're doing. We just do minus that. We do that, then we see that we are where we expect to be.
Oh, there we go. Now I'm just.
So this is the Atoll, which is north of the Seychelles. The very remote place. And there's no people living there, there's just a research station, and they went there with a group of students that were like the most successful students in environmental sciences in the Seychelles, and they took them for a week to Atoll and they sampled for EDNA. They sent us the samples, and it's really cool, and you can read the project's information on our website. There's some beautiful pictures.
Cool.
Then we have the event date, which is important. And with OBIS tools, you can check these things directly. And it gives you a message, it does not seem to be a valid date. That usually means that we have a wrong format for the anything, right? And so with this package, you can format to the red standard, which is the ISO standard. And since we have two samples, we have two dates. You can see we get down the right one for that. And now fixed. Then we can check the rest of the fields. So as I said, we have a few 8 required fields. And this check fields gives you the missing fields of those required fields. So in this case, we have a current status and basis of record. Now, of course, normally you don't have these in your data set because they are just something you need for the database and Darwin Core. But it's very simple. These are just defined values, so you can add columns with those values in your yeah.
Is it just those 8 essential ones that you can check, or can you check other?
I think it's just those. I'm gonna have to ask. I'm actually not sure. A good question. I would like to invite Peter on the Zoom. I don't know if Sophie, do you think you can send the programs? Yeah. If you just send the Zoom link. He said he would be available. Because he will have answers to all of these questions. Yeah.
This is our implementation guy already has some scripts to put file seek objects directly into the dislike format,
Great.
which you will again like share them as well.
Yeah.
We have that.
You see.
In the end, you just need to do it once.
Yeah.
The thing is that every institution works a little bit with different fields, different names, so I think the easiest is if you have it in your institution for your workflow, and then you can just run it. But we can of course provide these general purpose ones.
Yeah.
But a lot of like what the integrated publishing toolkit does, or the eDNA toolkit of GBIF, is it maps those fields, so that's the important step, right? To get those right Darwin core names to your fields. And that's something that's a bit hard to automate, because people use any kind of names for their columns.
And that's, I think, what Miwa's project, Making eDNA Data Fair, is working on, trying to map kind of what usually labs are keeping track of to terms that will then be mapped to GBIF and OBIS terms and MixS, so that we'll have like one thing we can go to hopefully and just match our data to and then submit it to different places.
Does that create another layer of names between, like?
Well, what I'm thinking is like, once it settles down of what names we should be collecting between Miwa's work and the NOAA template that Luke Thompson and Catherine Silliman are working on, then I can just easily create a translation of what our lab collects to those terms.
Yeah.
And if those terms, yeah, hopefully then have mappings to what OBIS wants, and so you just are matching to one thing instead of
Yeah.
having to convert between multiple different things.
Yeah. But then again, we still do have, there's 3 different terms, right, that then have to be mapped, but that's not difficult to do.
Yeah, I'm hoping like institutionally,
Yeah.
MixS and OBIS will be talking to each other. And like that'll happen in the background, so not each scientist is figuring it out. We can just say, okay, each scientist only has to match it to one standard, but then we'll have converters.
Yeah.
That conversation happening.
Well, I think within Miwa's project, she's connecting those groups together. So my hope is that that's happening, because I don't think that should be on the scientist's back. I think it should be on the institute's back, kind of to make sure there's converters between what we're asking scientists to do.
Okay, and then we had a few of these measurements that were, for example, sample size. So that was actually the amount of water that was filtered for the sample, and DNA concentration. And these we can then add to the EMOF table, so the extended measurement facts. And as I told you earlier, and seawater temperature. So we can add measurement type, you can add any text you want. Of course, we want to try to make it understandable, but then how that links and how that becomes machine readable, and so that everybody understands what you've done, is through those ID fields. So those are the fields that you look for in vocabularies, and then you add a URL to that vocabulary, and that ensures that these are all understandable across the board.
So if you have a different field you'd like to add, you just need to look up which NERC URL you need for that.
Yeah. So it can be anything, and there you so suggest new terms, and so, but it's a whole different thing, yes.
Do you think eventually like some of Miwa's terms will have unique IDs that you'll just reference in this section? Or will it always be like a NERC kind of term you're looking up, or?
I think it can be any vocabulary, so it's
Vocabulary, so-
possible, yeah, you can have a URL, but I think we recommend that.
Okay.
But we hope that Miwa's terms will become part of the extension, right? So that will, then you won't need to have this separately, but your ID will be your column name. That's at least mine, but of course, if there's something that doesn't fit, then it can. And yesterday we were also talking about, or you were talking about like fields that you couldn't map into Darwin Core.
Yeah.
So these things you can also add into field, I have to remember again the name, but there's a field basically for anything else. And you could add it, for example, in a JSON format. So if there are fields that you want to keep in your institution, these are super important to us that they are in our data set. You can add them there. Yeah, then it's of course not searchable, it's not necessarily comparable to other data sets, but at least you will have that data available. I don't know anybody else, but they have to know what you're doing, understand this. And then the sequence data. Are we good to move on? So we can read that FASTA file, and then you have your sequences here. Characters. We use this package called biostrings. You can add that to your table. And then you have all your information. Yeah, this is the DNA. Then finally, we have the metadata information. So in this case, we have mainly primary information, references, and library layouts and sequencing platforms. So these are, for example, things that you would have from your SRA submission and your mixes tables that you can then link here as well. But there's many more.
I guess I had a question, kind of earlier we linked a protocol under a field, and often you have separate protocols for different steps of the pipeline. Do you just, is it all gonna be under that same field and you do semicolons or?
Yeah,
Yeah,
exactly.
okay.
I think that's what you would do now. I don't know how, does you wanna have separately?
I got
Yeah, like maybe that's something we could add, protocols for different steps.
Sure.
And now we do the same for the DNA file that we did for the occurrence file. So the occurrence file was basically linking the sampling information and the taxonomic information, and the DNA file is linking then all this information on the DNA.
So this kind of metadata could maybe be pulled out from VLOOKUP.
Yes.
And what are these OBIS eDNA reference databases? I can't connect unfortunately, but is that like just a list of common ones or is OBIS that is its own?
We don't have our own databases, but which one are you referring to?
For RefDB, it just has a URL. A GitHub, I have. Right,
Yeah, that would be I think our protocol for making the reference databases. So we are using this package called CRABS to extract reference databases from NCBI for a specific biomarker. Specific taxonomy.
do you call it crabs?
CRABS, it's not our package.
Right,
It's a
well, there's a very similar one called
Yeah.
Crux.
Yeah.
Oh, yeah,
It
yeah.
just sounds like it does the same thing.
Yeah, right, yeah.
Okay.
So this one starts with an NCBI text search.
Oh, okay,
Yeah,
that's a
it's
different,
all
yeah, this starts with
sequences.
primer sequences,
Yeah, and then you can do a PCR on those,
gotcha.
and then a PGA, and then you add your names, and then so you make like a.
Interesting comparing that to specs.
Yeah, it's not like there's one extra filtering step with the taxonomy or keyword search, but.
So what's called Crux.
Or I guess R Crux is, it's the R version, but I think that's like the only. version.
Creators of like Anna Kappa and that part of that same kind of family.
Is that what you use?
It's,
I've been using it a lot, but
yeah,
not always.
I just check, there's some coffee and stuff outside. Do you wanna have a quick break now?
Yes, we have one step, and then we can have a break.
Can I also check that this SAC members, does anyone desperately want to stay here until the end? Okay.
Yes.
okay, I can ask questions later.
You wanna stay here?
No, it's
You-
okay.
you'll go. Yeah, okay, so when you're ready to break, we'll have coffee and then, SAC.
Yeah, so we just have the final step, which is writing the text files and compressing it into the Darwin form. So we take that occurrence file that we made, we combine our different files into the MOF, the extended measurement artifact, vocabulary terms, and then our DNA file separately. And then for the archive, we add some more metadata here. And you just archive it with writer's package. And that's it. Then we have an archive and it can be uploaded to the IPT and it should directly be mapped, so you don't need to do much. Because you have all the right terms. Amazing.
Thank you.
Thank you.
So for those, we can do a break now. Is there any questions online?
I guess I had a question at the very final step.
Yeah.
Is this actually uploading your data to GBIF then, or OBIS?
No, this is just making the archive. So you just
Okay.
make the zip file.
Okay, yeah, it was querying the internet, or I just noticed
Oh.
it doesn't work for me because it says it's failed to connect to
Okay.
gbif.org.
Probably.
Maybe something up.
Yeah. Probably.
Oh,
That was sort of my question too, so then we have like files and then we talk to our node person.
Right.
And then we talk to our node person
Yes.
basically and say, here's a bunch
Yeah.
of zip files.
And maybe they will let you just upload this. Yeah.
Should I just ask you?
Yeah. So all the node information is on our website as well, but I can.
Here's the deal.
Oh, really?
for this final step, when we're able to do events in additional to occurrence, then it would be kind of creating something additionally in the core part, or?
You will have a separate table. You will have an occurrence, an event core table, an occurrence extension, a DNA extension, and an email.
Okay, so then the final step will just put all of it together again.
Okay, if there's no questions, so we can go for a break, also online, we're gonna go for a break for 10 minutes or 15 minutes. And then I know the SAC is leaving, but, so we have another markdown for DNA access from OBIS, so if you're interested, you can just run that through. on your own. Let me know. And we can look into the GBIF too, depending on what. Hi everyone online.
I'm really excited when she's here, cause I was like, no, we can do
That's why I was
So I was
myself.
like, experience, we're like, we're in different countries, so it's like.
We're just gonna get started again. With the next step. If does somebody wanna look at the GBIF tool? Maybe we can do this and then I can ask for the other steps but if you want to try that out, cause that does the mapping in like in a visual way.
Is it
It's not gonna take long, is
Okay.
it? So we might as well.
Sure.
No, yeah, but maybe we'll do this DNA axis one first.
Yeah.
And then we can look at that.
They ask for it, they want you to say the right words. I really have no internet, for real.
Oh, you're very small.
Yeah.
Oh, it's a very big room.
Is the GBIF tool in the repository that we cloned?
Yeah, there's
Oh.
a link, there's a link to the
Okay. Well, I'll link to a website.
Yeah, but it worked for me yesterday.
I think now I've like completely lost it.
Oh.
Like I don't even have the limited thing anymore.
Oh.
But let's see.
I can run it through, but let's do this one first. So in this markdown, we're looking at how to access data from OBIS and how to access DNA data. It starts a little bit with just how to use our markdown. We don't need to go through that since we've already gone quite far. Here we have the OBIS tool.
Cars.
So first of all, we can collect all the information of what DNA datasets are in OBIS by using this has extensions DNA derived data. So this way you find all the information on the DNA datasets. And what we can see is that we have now 52 datasets. And here you have all the information, so the titles and what they are. So you can see we have a lot of marine microbes at the moment.
Can you go back up for a minute?
Yeah. So these are mainly Australian data sets. We also have the nodes here. OBIS Australia, OBIS UK.
Sorry, can you go back up in the code?
Oh, sorry. Yeah.
Right, so
So this is
where are you defining the URL? Yes.
so it's just the data set.
Oh.
So this is
okay,
querying
right, I
OBIS.
see. Okay,
or-
cause I'm getting null for.
No, that might be, it could be an internet.
I got the days. Okay, yeah, that's all right. I'll just watch.
Yeah. There is also this one, it's in the solutions folder.
Yeah, that's what I.
I
That's the one that's null. Okay, I might just have the wrong. In the solutions folder. Okay. Let's see. Directory.
So you get information on all the nodes of records, which node has submitted this data, the geographic range, and guidance. And in this next step, we're looking at where these DNA datasets are in the world. We take all that extent information. And here is our current range of eDNA datasets.
Does it generate the polygon itself, the coordinates you put in?
Yeah. It does, I don't know what this exactly does, sorry, it's not my.
I was just thinking with our AMT groups, we'll end up with just a massive polygon.
Yeah, I mean, these are massive, right? I mean, it's, yeah. But it's at least to show that we do have something. And so then if we want to fetch data sets from that list, so you might want to look into something that you're really interested in. Now, in this case, we're looking at the Pac-Man data sets. So I'm just querying the title names for Pac-Man and selecting the ID. And then getting all that occurrences from that ID. In this case, I'm selecting only those which have are recorded in the RIMS, so are invasive species detections, or are in the invasive species database. So that's just to keep the number of occurrences small. Because we have, I don't know, 300,000 occurrences. So in this case, we get a small subset like this. And if you want to look at what that data set is like. So you can see that it has basically everything that we did in the last step, so brought together. And this is the way you can get everything. So organism quantity, scientific names, the different taxonomic levels, and their IDs, locations, and so on. And just to mention, at the moment, what we're doing is we're adding our bioinformatics information into this identification remarks in our own formats. So this is, of course, because the extension doesn't have such complicated ways to put the bioinformatic information, and we have these multiple annotations and things. So we made this JSON format for ourselves with identification remarks. So you can do these types of solutions for your own data sets as well. Or we'll look into MUAS fields to see if that's something useful for recording your purposes. Then we look at the taxonomic composition. So we're just looking at the phylum level, number of species and number of records. And you can see an overview of what type of organisms we have in our data sets. The DNA-derived data extension is nested inside this occurrence table. So we need to unnest it. If you don't have access, if you're not able to get that occurrence table, this information is here in the solutions. Continue from here on. Then we can start looking at that information in the DNA table. Target gene, sequence, DNA sequence is there. So we have those PCR primer names and that makes us information on the sequencing platform. Finally, the DNA sequence as well. We look at a bit the composition of this. So what we see is we have two different target genes that we've looked at with Pac-Man data sets, 18S and CO1, and we have some thousands of records for both of those. And now we'll just go through a couple of examples of how we can work with that data, what kind of things we can do. So I'm just making a subset of our data first. So we have 100 sequence records. Filter for only the CO1. And then we can read the sequences into a format that we can work with in R directly. I'm running through this quite quickly. I don't know if everyone okay with this. Yeah, it's to demonstrate how we access data and how we can work. We can align those sequences, so we have 34 sequences. We can visualize the alignment. I didn't find such a good way to do this yet, but this shows a little bit if your sequences have aligned correctly, so it's important to just check the alignment. Looks okay. And then we can make a function. But you can imagine that you can collect data for a specific species, for example, for in a specific location. It doesn't have to be just from a data set, but it can be from many different ways, combining data in OBIS and coming up with these DNA data products from that information. So it becomes a very powerful way of exploring data, right? Once you have all that information in your database. So you can access data with R, but you can also access it through the mapper. If you haven't checked that, just go through our website and into the mapper, and you can select for the extension again. You can see all the data sets. Just do what we're doing here. And then if you download them, you will have the separate tables for occurrence and DNA extension, but you can just combine those with the ID. Now we have also a new prototype, which is the sequence search tool. This is really, really new. So Peter shared this with me yesterday. They're the first ones to try it out if you want to try. Of course, we don't have. So basically what it does is you can have your sequence, do a search to find your identity threshold, and then it'll find you occurrences that's have that identity threshold for your, and then you can see. It's also a nice way to check how good your annotation is or how good the annotations are. So for example, here we have quite a distribution of different annotations. So this will allow you to do some quality control. Another example.
That's cool.
Yeah,
Can you limit the search to certain geographic regions or?
I think not yet. So this is really the first prototype of what we have. But I'm pretty sure that's, yeah. We can do that later. But I mean, if you want to look at just search for the extension, like search for data sets with DNA extension in your area, that you can do very easily with the VAR package. So this is just the sequence search, but for the other searches, you can do all those.
So you could filter, if you get all the records in OBIS, you can filter by geography.
And even in the access, you can define the geography that you want.
In the access.
So in the R. So here, I think when you're looking at data sets or.
Right.
For that, yeah. You can define a certain polygon or certain species that you want to look for. So it doesn't have to be specific defined data set. You can do all kinds of searches that you would do in the mapper as well. You could define an area and get all the data that's in that area. Cool. So for example, here we have a hydrozoa in Australian data sets. We have a lot of data sets in Australia. And define 100% matches we have in this area. Maybe this is one group that has been doing this. With more, we get some more interesting sequences, and you can look at how they're currently have been categorized.
It's interesting to see.
So that's it for this one. It's a very quick run through. If you want, we can look at the GBIF tool now. Would that be?
Sure.
So Let's see, I think it was in. So you do need to make a login for yourself. And then you can go to new data sets. So this workflow is basically what we did in putting all the tables together that we had, the different motif table, taxonomy table, sequence table, and cluster. Map those terms, process that data, and then you can review, add metadata and export. And I think it's not yet connected, but in the future you'll be able to publish as well, especially you have to be connected to a node, but you would be able to directly publish through this tool. So it's supposed to make things easier for the user. So if we want to just run through this, I'm gonna put here OBIS training. And you have all these files here in this is the correct place. So I'm gonna put everything except the metadata. Let's see if I can upload, and then you just have to define what you have. I did have some issues with this earlier, so let's see if. So you upload, and then in the next step, you start mapping the fields that you have in your datasets. So this is what we did, except that you will do it, it's a bit more guided the way that you do it. More visually appealing and yeah, an interface to doing this. And it already populates what it can recognize. So from the taxonomy table, it, for example, does recognize what we're looking at.
Is the output from that a zip archive as well?
Yes, I think it gives you the tables. I think you can get the zip archive.
Okay.
And you can also get a bio formats. So there's many different formats you can get from this. Or it can be directly published as well. So now I'm just mapping like what we did in our markdown. I'm just gonna map the fields as we had. So we had latitude, longitude, target gene, I don't think we had, but we can add value here. So this is important because we can actually do taxonomic annotation with the user tool. Here, guy. And we can add other ones as well. We do, for example, locality. Name. Feature remarks for our parents' area name. I mean, you can choose these later or look at what we did in the R&D. Area uncertainty is coordinate uncertainty in meters.
So it's choosing the fields or like the column headers from the table you uploaded, the table you uploaded, okay.
So it's really just, again, mapping those names. The rest are not, so this would be the current score. Then we have the taxonomy table, which also goes there. And then we have facts. And here it already retrieves the DNA sequences from the FASTA file as well. So it automatically does a few things that make it easier for you. If you have the correct names. And then here, I want to do. All right, this is for the taxon. But then you can add the extended measurement or fact again. I don't think there's yet the ID fields here. Okay, and proceed. And so here you have either just processing the data, so that is making the tables, putting them together, or you can also assign the taxonomy, and I tried this out, and it doesn't take too long, so we can do that, but I'm just gonna first do it without, and then maybe we can go back and also assign the taxonomy and see how it goes. So this was just the first 100 sequences of our files, so it's a very, or first 500 sequences of our files. We have 374 taxa, that's the ASVs, 750,000 reads, singletons, and it gives you a few of these like overview of what your data looks like. I think it's quite handy. And then it'll give you a review. So it's saying we have invalid coordinates, so of course we would have to fix those. But it does also give you an overview of your taxonomic composition. And so now we only have 2 samples, so these are not that interesting. But it gives you some kind of information here. Then you can add your metadata. There's a lot of fields that you can include here. This looks very similar to what the IPT has as well. So this is the normal way that you would publish through the IPT. It collects all that information, that metadata information together. And then you can export and create a direct archive. And you can publish. So now it goes to the test environment. Sorry.
So easy. Oh, I just
Yeah.
This is the.
It's really nice.
Yeah. So these kinds of things are in process, but I did look into it yesterday. I think if we do a process data and assign taxonomy, let's see if it starts. Start. Can't redo it, but I did do the assigned taxonomy and I got quite different results. So it had a lot of insects, which I think it comes from those unknown sequences. That it probably somehow had fixed in there, so I would be careful with this assigned taxonomy tool, but it's a nice addition and probably something that can be developed. Maybe you can try it out with your data.
Is GBIF, are they aware that the assigned taxonomy tool is so inaccurate?
I don't know if it's so, and it's just because I think we also had them at some point for the. I noticed that I had a lot of insects in the unassigned reads that became, for some reason, became insects. So it's just something that we need to discuss.
What's your question?
Saara was saying that the GBIF assigned taxonomy tool wasn't it was assigning insects to reads, which are, it's nonsensical for marine data.
Yeah, but I get a lot of insect annotations for marine data
Yeah.
for CO1.
And it might be because you're using a curated reference library through your bioinformatic pipeline that doesn't include insects, so it classifies those as unknown. So, but if you're using just the GenBank as a reference library, then it will I don't know.
I still get, I mean, always get a lot of insects in my marine data, and I've tried different databases and looked manually at some of the sequences, and
Are they insects? Like, do you?
so some of them could be, you know, if we're coastal, and so there could be flies and things in the surface waters. But I think a lot of them are just organisms that are not in the database, that haven't been sequenced. They're weird little ocean bugs that we don't know anything about. That's my guess.
They could be symbiont and all sorts of, but the closest
Yeah,
related thing could be
totally.
terrestrial.
Exactly.
So, but if you have your cutoff at 97%, it shouldn't give you like a different family or different order to what the sequence is.
I've still used strict cutoffs and got flies. So
Okay,
So some of them might be
Some of
real.
them might be real flies and stuff, but having, say, 20 insect species would probably, I don't know if that's a real signal or just something to do with the way that you're assigning taxonomy.
Gotta also remember the regions that we're looking at are highly conserved and insects and arthropods.
And if
Quite
you
often
do,
I've done
yeah,
crustacean work where we've mapped to Drosophila reeds because that's the closest we've got, so.
and if you do any sort of LCA, like one workflow I've been using recently is just a blast against NCBI and then an LCA to find the most likely, like take your top hits and do an LCA, and then if it's really nothing in the database that's closely related, then I think you could easily get insects. Because insects are massively represented in NCBI, like way overrepresented, so, yeah.
Right, yeah. So that would explain. I'm doing this again, so I'm gonna assign it.
Just the insects.
The other one I get a lot, actually not for Amplicon data so much, for shotgun data is Magnoliopsida plants. Also, for some reason, tons of them in NCBI, they're like weirdly like of interest for some evolutionary reasons, so they've done a lot of sequencing on this like family of trees. Yeah,
Pretty trees in Magnolia.
beautiful, yeah, not in the ocean.
Yeah.
Yeah.
Yeah, so now if you, this is the what's GBIF gave me, so all of this is insects.
Wow.
Yeah, that's.
But I'm not sure if there is this kind of.
Elements,
This was the
dipped
thing
around
about the first
Dip
one.
into fly.
So yeah, definitely something we can.
I don't think it's a GBIF, well, yeah, we can ask them, but
Yeah,
it's,
yeah.
I've had those results when I haven't gone anywhere near GBIF, so.
Yeah, no, me too. It's not only GBIF, but then maybe they should be aware of that.
Yeah,
that
true.
It can kind of wrongly classify things that we shouldn't really give classification to.
So when you assign taxonomy here, you literally just tick a box.
Yeah. Yeah.
Yeah, I kind of get that you've not made it open to people to tweak things because you want it consistent, right? But yeah, it needs
Yeah.
further tweaking, doesn't it?
Yeah, I think it's just their first prototype to see, and they're looking for people to give feedback, so. Right. So again, you're special people you have access to now, these things, it's not open to everybody.
Oh.
Or it hasn't been shared that, I think.
But what she's saying is be nice and don't make them cry.
Always nice.
No, I wasn't saying that to you, but it's like, this is their first open
It
platform.
looks promising, but
Does.
obviously, yeah.
Yeah,
Yeah.
but these are the kinds of things, right, the tools that everybody is looking for, like, what are the tools that will help us format the tables that we have, or format a phylocentric object, and I think these kinds of things, yeah, if they're helpful. But then again, if you have an institution that does a lot of work regularly, then maybe this is not the best solution. Rather, you have your script where you do your mapping and then you can just always run it in the same way. You have certain workflows.
It's definitely nice for making it accessible to people who don't have that knowledge of it.
Okay, that was it.
So if we were to upload data to GBIF, that would get pulled into OBIS at some point,
So
right?
there is a checkbox that says submit also to OBIS. You do need to have a node that, or you need to have like an IPT instance for both GBIF and OBIS.
Okay.
So you will need to be connected with a node that's connected with an OBIS node, but no, I think your node can help you with that. Like that's any way you have to go through your node when you're submitting data, so. I think you can figure out.
Mhm.
Why is it optional to submit to OBIS? Wouldn't you want everyone to automatically?
Yeah, I think it's, yeah, it's big, I mean, if you have a terrestrial data set that you add to GBIF, then you don't really care. If, yeah,
Go the other way
yes, also the other way, yeah,
by default, or is that also a?
it's also a check. Decision you make.
Can you go back in and edit that? They actually, I want to.
I don't want it. I'm not sure about that, that's a good
Yeah.
question. Once it's out there.
With the data flow, but.
Right, is there any other questions or things that you would like to see in another training? If there's any questions online as well. Is there any extra step for the quality control for the taxonomic assignment or? Yeah, we did talk about that a little bit earlier, so I think at this point, I think everybody's doing their own. So we don't have any OBIS workflows for checking that. We are doing a lot of manual checking with OBIS, and especially in the eDNA expeditions, we have the local management is going through our species list to identify which ones are really local and not. And I think that's still the way, you still need that local knowledge of your community, of your species, to be able to really confirm those assignments. We do several different assignment methods to look into that, and also look at the species distributions and thermal ranges to kind of give a confidence score for those assignments. So there are certain ways that we are doing, and I think that's actually maybe a nice thing to do, like quality control of taxonomic assignments after.
It's a huge issue.
Yeah.
and everyone struggles with it.
Exactly.
And there's no like perfect answer yet. But yeah, there are some things you can do to make it a little
Yeah,
more robust.
That would be a nice note on our gathering everyone's knowledge and experience with it. and
Yeah.
Communicating that to people. These are things to consider.
yeah, I think so too, actually, it's a good network for that. And see how people are doing that and making sure that what they're putting out there is okay.
Yeah, one thing I was wondering about for either OBIS or GBIF, we get a lot of contaminant sequences in our data set, especially like 12S, so always human, usually cow, pig, things like that. Should we be uploading filtered data that we have removed like definite contaminants? And then we have like possible contaminants, like fish that may or may not belong there. So,
Yeah.
you know, when we have our sort of QC processing for final data, we try to do some of this quality control. Is there some like minimum amount we should do, like definitely get rid of humans or whatever?
Yeah, that's what we're doing. So with
Okay.
our eDNA data sets, we are getting rid of those contaminants.
Yeah.
Just filtering.
Okay.
Um,
And maybe you could put it somewhere in the notes or something,
Yeah,
like, or some method metadata there, it says like filtered for
Mhm.
X, Y, and Z taxa.
Yeah, indeed, yeah. Yeah, I think that's the best way. I mean, otherwise they do get dropped because they're not gonna be marine.
Oh,
taxa,
right, sure, yeah,
So they
okay.
will be dropped. But it is a problem if we get, for example, some kind of plants in the middle of the ocean. I know there's these kinds of issues that have come up. But I'm sure we will have these things come up more and more, the more data that is added. So yeah, we have to come up with a quality control for those. But first and foremost, yeah, terrestrial taxa are drops. Right? Thanks everyone online as well. If there's any other questions or comments, let me know. Otherwise, I think I'm gonna close the session. Is everyone happy? So there's a question from Mariska. Since it's only getting scientific names from worms, there are cases where the names are not updated nor listed. Yeah, so that's in these cases, we need to contact worms and figure out with them what's the best way to do it, if they can add it, if they can update it. So this is something that's, it has to be from their side, so this is not something that OBIS is doing, but you can always add your the name that you want in one of these fields, so either in the verbatim identification and maybe an ID from the other catalog, you can add that to the taxon concept ID. And you can still have your scientific name that you want in the scientific name, but your scientific name ID should be the closest you can get in worms. So I would go like a level higher, maybe the genera you can put into the scientific name ID, for example, at this stage. Or contact worms and see if they can add that. All right. Thank you, everyone. Don't hesitate, you can send me an email if you have any questions, or look at the other contacts from the OBIS website, so also the notes, and you can ask them directly for any questions or if you want to add your data.
Thank you so much, Saara.
So thank you. Thank you, everyone.